Yeah, so thanks Sam, and thanks everyone for the invitation, to the Soil Science Society for the invitation. Today I'm presenting on our GRDC project methods to predict plant available water capacity. And I had to have a picture of my truck because I love my truck. Now, plant available water capacity is used for a lot of things in agriculture. Growers and advisors need advice on plant available water and plant available water capacity in terms of millimetres of water available for a crop, so they can make decisions on, do I sow a crop, what do I sow, when do I sow it, what variety do I choose, when do I fertilise it, do I run the sheep on because I don't think it's going to yield this year, it's going to run out of water on me, do I bale the crop. Almost every management decision that you make is based on plant available water capacity. But we can see from this image here, this is a management unit down in here. These are all paddocks. There's not a lot of available soil water characterisations to throw into a model like yield profit or APSin to do predictions and make those decisions. So we're trying to come up with ways of how we can spatially extrapolate these soil water buckets and make them more useful. And in our Hard and Young study area, which I'm going to focus on, that's almost the extent of our soil water bucket network. So for the project as a whole, we had case studies in the Central Darling Downs, Western Downs, Central Highlands in Queensland. New South Wales, we had an area in Liverpool Plains, another in the Macquarie Bogan, Moree Plains, Central West of New South Wales, out around Condo, Lake Jellicoe. But I'm going to focus on the Hard and Young study area because that's where we had the most data. And it's closer to here. So, first step of the process was to do a lit review. Very extensive job, but we didn't just do the northern cropping zone for GRDC, so that's all the country from the Murray River to Queensland is the northern zone. We did the western zone as well, so that's all of Western Australia, and the southern zone, so that's South Australia, Victoria and Tasmania. So we did the three big reports, and the first actual infield bit of work we did was a reconnaissance soil survey where we just evaluated soil materials down strategically selected hill slopes to get an idea of soil material distribution, how that might relate to PAWC. And we looked at the lines of evidence that sort of go with that to try and help us make a decision on which soil water bucket to use for yield monitoring or for making those crop decisions. So then we looked at how we pull it all together. Step two was to try and validate our estimates and see how we went. We ran into a bit of trouble because we were doing it in 2018, 2019. A, because we couldn't measure crop lower limits because there wasn't much of a crop. And B, because when we were trying to do drained upper limit, we're asking our growers to provide us with between 5 and 10,000 litres of water, which they just didn't have, and we thought, that's a bit rich, bit too much of an ask. So we came up with another process where we interviewed a whole bunch of key advisors in the area to try and evaluate our methods to see whether they thought, just to see if they passed the pub test a bit. Well, it was a little bit more. Did that pass the pub test? To see whether they passed the pub test and whether they're likely to be useful, and how that might roll out into future extension programs, etc. So our Hard and Young study area is the area you can see with this colourful map here on the left. We're in the southern tablelands of New South Wales, about 650 mil rainfall zone. The whole area was underlain by the same parent material, and that was important because we wanted to try and limit some of the soil forming factors that go into variability of soil materials. And the other key point to note with all this is that there's a big drainage divide here. I've just roughed it in by hand on this map for this DEM. But that's the drainage divide between the Murrumbidgee catchment in the south and the Lachlan catchment to the north. And that gradient has a big impact on soil material and soil forming processes, and that drives a lot of our thinking of how we extrapolate soil water buckets. It's the geomorphic processes that control the soil material distribution and in turn drive what soil materials you'll find in what place. So that's the logic behind it all. That's about all we need there. Oh yeah, just geographically, Young's just here, Hardin's just here. That's Wollongbean there, but it missed out because it's in a different rock type. It's in the Kulak serpentinite belt. Oh yeah, just a point of notice, that green polygon there is pretty much along this catchment divide, and those soils in that landscape are aeolian soils that were blown in post last glacial maximum. Richard's a good man to talk about Parnas soils. He's done a lot of work in that space. But those soils in that unit are usually very thick, deep clay soils, very big soil water buckets, and that's why we see a lot of cherries grown in that area too, because it's high value land. There's varying amounts of that Parna material across the whole area, but the erosion rates dictate where it actually sits and whether it's actually stayed. So there's more of it in this northern part of the catchment than there is in the south, because it's dominated by erosional processes rather than depositional processes that are happening. So the activity gradient down there, it's highly erosive down here and very slowly erosive there, because the difference in elevation from here down to the Murrumbidgee, which is just down here, Dugong's about here, the gradient there of elevation is much more than the gradient of elevation between here and the Lachlan River, which is, I don't know, 100 k's north of there. Well, not quite. I can't remember. It's a fair way anyway. So some of the information we use to inform our decision and our selection of plant available water buckets. That's our scoping report that we did, our lit review for the northern region. We use the soil landscape information from eSpade, that's out of the New South Wales Department of the Environment and Heritage. We use terrain data from various sources, GA and others. Elvis is a site that we use. We use the gamma radiometrics, which are a map available through Geoscience Australia. That tells us a lot about erosional processes and the distribution of that Parna material. We use geological maps from New South Wales Government. We use some expert knowledge that we developed in previous project to come up with a conceptual model of plant available water capacity and how it's distributed in the landscape, and that's a good tool that we use. We've used the Soil Landscape Grid of Australia. Brendan's going to talk a bit later about digital soil mapping, and this is really his domain, but that's a national coverage of soil properties. That's also freely available. We got some data from the Department of the Environment on, I think, soil ESP and EC, which have helped us modify our soil water buckets when we might have a subsoil constraint. We also used profile data that was available. By the way, I've got 4 slides at the end of this talk that's got a whole bunch of links, resources, data, anyone wants it, it's all there. So we developed a 5-step process to try and estimate plant available water capacity from what's actually in our study area. But we had to really think long and hard, how useful is that to an advisor or a grower on the ground? I mean, how are they going to interpret what's in those reports? You sort of have to be a soil scientist to unwind a lot of what's written in one of these reports for the soil landscape unit. So for a non-soils agronomist, for instance, that's a bit of a challenge. So we had to be very mindful of how we put this data together and how we sort of work that process. We had to put our outputs in formats that were going to be, accessible. So we've used our data access portal. and we had to have it in a user-friendly format, like a GeoTIFF, which is pretty easy to use in a lot of different applications. And we had to have some spatial reference, so a grower can work out where they are on the road and what soil type they might be on and what data set they're looking at. So that was very much in the back of the mind. I've got this thing here because we really did throw the kitchen sink at the Hardin Young area. We used absolutely everything we could get our hands on to come up with these results. We even generated some digital soil mapping estimates of available water capacity and plant available water capacity to be used in this process, and they're available for all of the northern cropping zone, not just Hardin Young. We also developed a new terrain analytic that we've published, and we think it's going to be quite useful into the future. It certainly helps a grower make decisions about where they are in the landscape and where that position relates to a topo sequence diagram in a soil map unit description. So this is a basic summary of our 5-step plan, our 5-step process. The first step is to identify where we are. So this can be done in the office, step 1. Have a look, you know where your known point is. Have a look at what spatial data is available. Pick out that terrain analysis that we've made available. Have a look at the soil mapping units. And we've also, that's another thing we did, was we amalgamated a number of soil landscape units from that original map that had 8 units into another map that only had 4 to try and make this process a little bit easier. And we've used we're proposing to have a look at the gamma radiometrics in a fairly simplistic way. The second part, and where's Cliff? He'll love this bit. Second part is the Dale Kerrigan part of this episode. You've got to go and dig a hole. And Cliff, you'll be very happy. You do have to dig a hole so you can understand what sort of soil type you find at your unknown location and then apply it back to these other resources. Once we've dug our hole and we've got some information about texture, structure and whatnot, we need to assess the conceptual model of soil landscapes and their relationship with PAWC, have a look at the DSM, and the next step then is to estimate PAWC from a table that we've developed. The final step in the process is to adjust the available water capacity or the plant available water capacity based on known subsoil constraints. So if that's EC, or whether it's ESP, if it's sodic, or whether it's soil depth. And in our process, soil depth is a big limiting factor on PAWC, particularly in that southern zone. So this is a bit of an expanded version of what happened in that previous summary. First step, identify where you are on the map, pull out the report, have a look at the landscape diagrams and the descriptions of soil material. If it sounds all right, maybe you can go with something that comes into the table with that soil landscape unit. This is step 1 continued. So we also said to have a look at the gamma spectroscopy, and the way we're sort of interpreting this is to say, well, if your site falls in the pink or white, it usually means you're in a very erosion-driven landscape, because that's pretty much in situ weathered young granodiorite. It's full of potassium. Potassium gives you a big gamma response, and that comes out as pink or white in this ternary issue. Conversely, if you're in the blue or the bluey-green terrain, that's where we've got a lot of that Pana soil. And the Pana soil effectively masks out the effect of the underlying geology, because the Pana has very little potassium and gamma response in comparison to the granodiorite below. So in those areas, you usually got thicker soils, and that's that transport limited landscape that's often in the northern area draining towards the Lachlan drainage divide. Another option is to have a look at this modified map and see where you fall, and if for instance your your site was in one of the variants from the previous map, you could have a look at the main description for the main polygon in this map and see whether that's a better match for your site. I think that's it. Now step 2, this is Dale Kerrigan's place. We look at the textures of our soils, both surface soils and at depth, and we highly recommend going to at least 1 metre or the rooting depth of your crop. And that can be a very big story if you talk to John Kierkegaard about his site at Iandra. But if you can dig 1 metre hole and assess the textures of your soils and also aspects like structure, anything that can affect plant available water capacity, carbon content, those sorts of things. Our area is full of texture contrast soils. The red chromosol is probably the dominant soil type for the whole area, although we do have red dermasols in some of those Parna soils, and we have red candasols as well. In the in situ soil environments on the granodiorites, we have red chromosols at the top of the slope, and they go grade through to brown chromosols and then brown or grey soda-sols in the flow lines is a typical topo sequence. So once you've had a look at your soil types, have a look at the terrain position. So this map has crests are in red, upper slopes are in that orangey colour, mid slopes are the sandy colour. We go into light blue on the lower slopes and blue in the drainage depression. So automatically, because we know that soda soils form in those lower landscape positions, that's a likely spot you're going to find it, and that might be a subsoil constraint. So you're starting to think about those things already at this point.
3 minutes, Mark.
Holy dooly. Okay. And then you relate those back to the other mapping units that we've used. This is the conceptual model. So this is like the drainage divide. That's the material that's eroding quickly to the south. It's got a lot sandier material and shallower soil depths, and that results in a smaller bucket. This material here near the crest is a very thick soil water bucket, pretty much unlimited bucket, that one. And as we come down the slope, those PAWCs decrease with distance down the slope, and this one's inhibited a bit because it's sodic. Also, we pull out the value point at in the digital soil mapping and assess that as we go as well. Our next step is to assign a soil water bucket based on all that information. Now, we've only got 6 in the catchment, and we haven't got every map unit, so we've got to do our best by trying to match these descriptions, the soil landscape unit, all that data I've been talking about, to a plant available water capacity that's in that APSoil database. We've modified these to 1 metre to compare with our digital soil mapping. The results we find are pretty good from point to point. The relative amount of plant available water capacity is usually pretty good. The problem with it is the actual range is a little bit too narrow. So that means that soils in sandy soils or shallow soils have too higher PAWC estimate in the DSM, and soils with the unlimited thick clay soils that we see in those Parna landscapes, it's under predicting those. But you can still use it as a bit of a conceptual idea to help you make choices. The last step in the process is to modify the soil water bucket. Subsoil constraints like pH are usually based on what an agro would already know in that paddock. We know that the Hardin Young landscape has very little EC issues. We do have ESP issues with sodicity at depth, but they're very strongly correlated with this map. Terrain analysis can also help us modify a soil water bucket based on soil depth, and that's typically where we'd have to modify that estimate. We do have, if we're in a different, one of those other study areas, like in Liverpool Plains or in Western Queensland, we can modify PAWC based on a chloride value, and that model was also developed for that purpose. So what's new under the sun? I've already mentioned this. We think it's a good thing, and we're hoping to use it a bit more in the future, and we're hoping that other people might use it for both DSM and maybe in a precision ag setting. Also, the other thing we've generated as part of this process is a web app that actually drills into the data sets that sit behind it to allow a grower to sit there and pull these data out and use it. But we've only got it for the Central Highlands area. We could, in theory, develop that up for every one of our case studies. Last one. What's new under the sun? Now, this is a PAWC inverse modelling done by some colleagues of mine, Dee He and Enli Wang. They've got two scales in which they operate. They can use cropping yield results, time series, and then use this inverse modelling approach to predict plant available water capacity spatially. And that's worked out very well. These are validated against AWC points from the APSoil database, and the R squared's 60-something. And they've also used bioclimatic regional data to do the same exercise using APSIM to predict plant available water capacity at different depths in all of the cropping zones. So that's pretty SMEC stuff. There's a lot of future in that, in my view. There was something else I wanted to say about too. It's just dropped straight out of my mind. Yeah, that's about it. Righto. Sorry.